You are listening to Professor Megan Workman. Welcome to this lesson about plasma membranes. During this lesson, you will learn to describe the structure and function of the plasma membrane, including its key structural components, phospholipids, sterols, proteins, and glycans, which are a type of carbohydrate. What is a plasma membrane? This is a general term used to describe the membrane encompassing cells and membrane-bound organelles. The function of all plasma membranes is to control the movement of molecules into or out of the thing that the membrane surrounds, which is either a cell or an organelle. In this way, the plasma membrane acts similar to a customs and immigration officer, which might try to control the movement of substance entering or leaving a country. Some example plasma membranes are the cell membrane, which surrounds an entire cell, or a mitochondrial membrane, which surrounds a mitochondrion, the nuclear membrane surrounding the nucleus, the multiple plasma membranes of the Golgi apparatus, the membrane surrounding a lysosome, etc. The basic structure of a plasma membrane is said to be a fluid mosaic. Here I show some real-life pictures of an iceberg floating in an ocean, and under that a beautiful mosaic that is made more beautiful because of its component parts, not despite having a diversity of parts. In the same way that an iceberg floats in the ocean, certain structural components of the plasma membrane float within a sea of phospholipids. Here in the image of the plasma membrane, you can see plasma membrane proteins floating like icebergs in a sea of phospholipids. The plasma membrane is said to be a mosaic because it is not homogeneous. It has multiple different types of parts, each part serving an individual function that aids the overall function of the plasma membrane as a whole. The key components we will discuss today are the phospholipid bilayer, which is sprinkled with sterols, proteins, and a few carbohydrates mixed in. First, let's talk about the phospholipid bilayer. And if you haven't already, I encourage you to listen to my lesson about lipids in which I introduce this topic. So I'm going to go a little bit faster through this lesson, thinking that it is going to be a review for most of my students. Here you can see a cartoon rendering of a phospholipid. This is just one phospholipid. You'll notice that it has a phosphate head where there is a phosphorus atom attached to oxygens, and because of the electronegativity difference between these two molecules, they form a polar covalent bond, making the phosphate head of the phospholipid hydrophilic. Opposite to this hydrophilic part of the molecule, there are fatty acids. Recall that fatty acids are comprised of carbons bonded to hydrogens. Because the electronegativity difference between carbon and hydrogen is low, these create non-polar bonds. So the fatty acid side of the phospholipid is hydrophobic. When let's imagine dumping a bunch of phospholipids into a bucket of water. They will arrange themselves in this way because their hydrophobic tails will be repelled by the water. What they will form is a bilayer in which the hydrophobic tails are touching each other, leaving the hydrophilic heads on the outsides. Here's what it looks like when it's zipped up. You can see the hydrophilic layer in the middle protected on either side by these, I'll call them liners, of what becomes the plasma membrane, where there are hydrophilic phosphate heads facing the inside of the cell or organelle, and also hydrophilic phosphate heads facing the outside of the cell. So this allows a cell to exist floating around an aqueous solution like in a pond or in the human body. And it also allows the cell to have an aqueous solution on the inside with this thick hydrophobic layer separating the inside from the outside of the cell. Here's why the hydrophobic layer is perfect for making up a cell membrane. Imagine a cell needs to maintain its hydration status. So here's a bunch of little water molecules, and maybe because of the concentration gradient of water, they're trying to escape the cell. Maybe it's drier on the outside of the cell. That hydrophobic layer blocks these water molecules from easily diffusing. So while water molecules can actually diffuse across this hydrophobic layer, it happens at a pace that is so slow that the cell can still maintain its hydration status and continue living unperturbed. The hydrophobic layer slows it down. Thus, this form of the phospholipid bilayer with this thick hydrophobic core functions to maintain the cell's hydration status. It reduces the cell membrane's permeability to random acts of diffusion. Now we see the phospholipid bilayer extending around a sphere, which could be the cell, or maybe it's a membrane-bound organelle. To me, it looks like a basketball. Because of the form of the phospholipid bilayer and this reduction in permeability, or the passing of molecules from inside to outside or vice versa, it allows the maintenance of an internal environment that can be very different from the external environment of this cell. It is the phospholipid bilayer and its unique chemical structure, meaning that hydrophobic core, that allows life to exist. It allows cells to maintain the internal cytoplasm milieu, the internal chemical solution that it needs to perform cellular functions and to maintain life. That can be very different from the pond it's floating in. Another key structural component of the plasma membrane's bilayer is sterols. Sterols are another type of lipid. In animals, the type of sterol that we produce is called cholesterol, but other eukaryotes produce sterols that are incorporated into their plasma membranes as well. For example, plants produce phytosterols. You might have heard of phytoestrogen. That's one example. In the plasma membrane, the function of these sterols is to embed in the phospholipid bilayer, the way you see cholesterol doing here. They're the little yellow guys that have wormed their way in between their phospholipid neighbors. And in doing this, these sterols fit randomly between the phospholipids and cause the tails of the phospholipids to kink and crisscross one another. You can see that going on in the cartoon, and that is true to life. So when cholesterol squeezes in, it causes a compaction, and it causes some randomness in the crisscrossing of these fatty acid tails. And that reduces the permeability of the membrane. It causes diffusion to slow down. It also improves the structural integrity of the membrane. And by this I mean that it resists more force. So if I was trying to puncture a plasma membrane with a needle, it would not as easily be punctured because the sterols are present. Sterols also allow the membrane to remain fluid at colder temperatures, so it doesn't freeze as easily. And it allows the membrane to remain intact at warmer temperatures, so it doesn't melt as easily. This function of sterols, when they're embedded in plasma membranes, allows the membrane to remain functional in a wider range of temperatures. Another benefit of adding sterols to the phospholipid bilayer is it allows it to extend out and curve around the spherical shape of a cell or organelle without breaking. The next key component of plasma membranes are proteins. Those are the little purple blobs you see floating in the fluid mosaic model in the picture here. And there's three types of proteins in a plasma membrane that we will discuss. The first are called integral membrane proteins, which are those that are permanently attached to the membrane. That contrasts with the next type, peripheral membrane proteins, which are only temporarily attached to the membrane. They come and go, and they can exist on either the inside or the outside of the membrane. Then the third type are called glycoproteins. These can be either integral, meaning permanently attached, peripheral, meaning temporarily attached, but they have to have a carbohydrate flag, which you can see here, called a glycan. Let's examine integral membrane proteins first. Recall that integral membrane proteins are permanently embedded in the plasma membrane. So they are floating around in the phospholipid bilayer, and that's where they spend their entire life. They are able to exist in the phospholipid bilayer because chemically some parts of this protein complex are hydrophobic. These are the parts that are integrated into the middle core, the hydrophobic part of the bilayer, while other parts of the membrane are hydrophilic, and these are the ones that are located on either edge. So they associate with the phosphate heads of neighboring phospholipids. They also associate with the aqueous solutions on either the inside or the outside of the membrane. Like this one in the image, some integral proteins pass all the way across the membrane from one side to the other. We call this type transmembrane proteins, but not all integral membrane proteins are transmembrane proteins. Some only go part of the way through the membrane. Integral membrane proteins serve a variety of functions. It is not my intention in this lecture to explain all of their diverse functions. You can read about that in the assigned reading for this unit. But here I just wanted to give you a brief introduction. So I'll talk about three. The first is cell-to-cell adhesion. Here you can see an intercellular joining event where these are two neighboring cells. You can see the phospholipid bilayer of the one on top next to the one on the bottom. And they each have an integral membrane protein that is reaching out and chemically bonding to its neighbor. This is useful in cells that aren't meant to move around. So cells that are part of a tissue that aren't free floating, integral membrane proteins anchor them in place. The next key function of integral membrane proteins is cell signaling reception. So here is a cell signaling event with two integral membrane proteins, one in the cell below, and it has this little glycan flag floating up to the one that I've circled. The one that I've circled is the receptor. So you can see it has a binding site. You could also call that a docking site. And it is receiving some information from the cell below it. So it is integral membrane proteins that do the reading of cell signaling. A third important function of integral membrane proteins is transport. Transport refers to the movement of molecules from one side of a membrane to the other. Recall that the phospholipid bilayer, because of its hydrophobic core, reduces permeability. That means it reduces the chances that molecules will randomly diffuse across the membrane. This is great because it gives the cell a fighting chance at maintaining homeostasis, meaning that it's keeping its internal environment distinct from the environment in which it's floating. However, you may be able to think of situations in which the cell might like to import certain molecules like food or export certain molecules like waste. Transport proteins embedded in the phospholipid bilayer allow the selective movement of certain types of molecules either into or out of the cell. In this way, transport proteins are regulating the movement of molecules across the plasma membrane. This gives the plasma membrane an important property called selective permeability. The selective permeability of the plasma membrane is allowed by these transport integral membrane proteins, which allow the cell to maintain homeostasis. The next type of membrane protein are peripheral membrane proteins. Recall that these are only temporarily attached to the plasma membrane, meaning they can float up to the membrane, attach to it for a while, then release themselves and go somewhere else. Structurally, most are hydrophilic because they tend to associate with the surfaces of the membrane, either inside the cell or outside of the cell, meaning they are interacting mostly with those phosphate heads of the lipid bilayer, which are hydrophilic. Like this one, some peripheral proteins can embed a little ways into that lipid bilayer, but others attach themselves to integral proteins, meaning they remain on the outside of the lipid bilayer completely. So in this cartoon that I've borrowed from the Amoeba Sisters, the big purple blob is the integral membrane protein. Notice it's a transmembrane protein. And on top of that is the peripheral membrane protein wearing a little party hat. Peripheral membrane proteins have a couple of key functions that I'm going to talk about: biosynthesis and regulation. Peripheral membrane proteins are often enzymes that perform cellular work. They assist in chemical reactions that make new membrane components. So they can build new phospholipids, they can help produce new integral proteins, they can insert or remove cholesterols from the plasma membrane, they can attach carbohydrate glycans onto either lipids or proteins. And so they serve a maintenance function to keeping the membrane itself healthy. The second function of peripheral membrane proteins that I'm highlighting in this lecture is regulation. They are involved in cell signaling. They are involved in the binding or unbinding of hormones from a membrane. If you've heard of G proteins, these are types of peripheral membranes that are involved in transmitting signals. Peripheral proteins also serve a regulatory function for whatever the integral membrane protein they're attaching to performs. So here in the Amoeba Sisters cartoon, this peripheral protein with a party hat is either shutting off or turning on this channel or carrier protein that is the integral protein it's attached to. So by temporarily binding to integral proteins, the peripheral protein can regulate its function and therefore regulate the movement of molecules in and out of the cell or organelle. Peripheral membrane proteins also play an important role, like tugboats shuttling around non-polar molecules through aqueous solution. So think of a big hydrophobic molecule, like a lipid maybe, and if it wants to come into the cell and then travel to some destination somewhere else in the cell, it comes through customs at the transport protein. It's picked up by a peripheral membrane protein on the inside, and that peripheral membrane protein tugs it to its destination through aqueous solution, even though it's a hydrophobic molecule. So the peripheral membrane protein is assisting that movement of the hydrophobic molecules in the cytosol. That leaves the last of the three types of membrane proteins we will discuss, which are called glycoproteins. And remember, these are any membrane protein that is attached to a carbohydrate flag. They can either be integral or peripheral proteins. Here I'm naming the flag. It's a glycan. And the function of glycoproteins is signaling. Like this one, some glycoproteins are peripheral. They can be outside or inside of the membrane, with their glycan flag waving towards the outside or waving towards the inside of the cell. But others are integral glycoproteins. And in this case, usually their glycan is located on the outside of the membrane, doing some signaling to the world outside the cell. An example of an integral glycoprotein is the ACE2 receptor, where SARS-CoV-2, which is the virus that causes COVID-19 disease, docks in order to gain entry into host cells. So glycoproteins have these carbohydrate flags called glycans. And these are so common that they make up about 10% of the plasma membrane. 90% of the plasma membrane is made up of phospholipids, sterols, and proteins, but about 10% is these glycans, carbohydrates. That's pretty important, so now I want to highlight their functions. Glycans are these carbohydrate side chains that can be attached to either a membrane protein or a membrane lipid. So on the left-hand cartoon picture, you see the glycan attached to a phosphate head of a phospholipid. That turns that phospholipid into a glycophospholipid. So whenever you see this prefix glyco, it means that the thing has a glycan attached to it. It's been glycosylated. The cartoon image on the right you've seen before, that has a peripheral protein with a glycan attached, so it's now a glycoperipheral protein. Glycans come in complex structures, and they can come in a broad diversity of structures because it is a chain of carbohydrate monomers put together in this branching pattern, and they can be put together in an infinite number of ways. So there are multiple monomers that can be assembled in an infinite number of ways. This relates to the purpose of glycans. Because of this diversity, the structural complexity of glycans, each type of cell can have its own unique glycan. In this way, your body can tell the difference between a neuron and a skeletal muscle cell. In this way, your immune system can distinguish between a self cell, one that's supposed to be there, and an invading pathogen, which has its own glycans that are different from the ones all of your cells produce. So the function of glycans is signaling identity of the type of cell that it exists on, and it's conveying information the same way a passport conveys information about a person's identity and other attributes like their nationality. In this way, similar to a passport, glycans can allow cells or enveloped viruses, anything with a plasma membrane that has glycans on it, to gain entry access across a membrane. They can also play a role in evading the immune system or oppositely triggering an immune response if your immune cells either recognize or don't recognize that glycan. as something it should attack. And glycans can also initiate a cascade of events by signaling information to a receptor somewhere. Some glycoproteins that you've heard of are antibodies. So an antibody is a protein produced by the immune system in response to seeing an antigen for the first time. An antigen might also be a glycoprotein. It could also be a glycolipid, but antigens have glycans on them as well. So here, your immune system sees a novel antigen for the first time, and because glycans can be put together in an infinite number of ways to produce a broad diversity of glycans, the immune system can tailor an antibody to any antigen you might experience throughout your entire life. Antigens that scientists don't even know about yet, that haven't yet evolved, your immune system has the capacity to produce an antibody glycan to match that antigen if it comes in contact with it in the future. And that is an amazing function of glycans. An example of a glycolipid that you've heard about, but you probably didn't know it was a glycolipid before, are the cell surface receptors on your red blood cells that define your blood type. You're either type O, type A, type B, or type AB blood. It is your glycolipids, you can see them here in the cartoon, they look like little spikes, that your body attaches to the outside of your red blood cells. These are glycans, and you can see the component monomers, and they're arranged in different conformations, and it's the conformation of these monomers that are unique to each blood type. So you can see that the O glycan is different from the B glycan, is different from the A glycan. And then if you are type AB blood, you'll notice that you have both the A type glycan and the B type glycan. So that's just an example of a glycolipid that I thought a lot of students had probably heard of before and just never knew that it was a glycolipid. I'd like to share one more fun fact about glycans, and it's related to a hypothesis that is attempting to explain the epidemiological observation that red meat eaters suffer higher levels of chronic inflammation. And this makes us, I eat red meat, more susceptible to heart disease. The hypothesis has to do with a glycoprotein called nu5GE that is produced by most mammals, but not humans. So when I eat mammal meat, I'm exposed to this glycoprotein, and in my body's quest to be efficient, the human body is able to uptake cellular components from food and recycle them into the new cells that my body builds. So this nu5GE glycoprotein is recycled by my body and incorporated into the plasma membrane of my cells. So if you took a cell sample from me, you would see some nu5GE glycoproteins on my plasma membranes that would not be present on the plasma membranes of vegetarians or people who, in a committed way, stick to chicken and fish. This is hypothesized to be a problem because nu5GE is not something my body produces naturally. And so my immune system is very smart, and it detects this glycoprotein and produces anti-nu5GE antibodies and actually attacks the cells that have this glycoprotein on the membrane. So my immune system attacks my own cells because I have incorporated these mammalian glycoproteins that otherwise wouldn't be there if I did not eat red meat. So that's kind of interesting. So now you have learned about the structure of plasma membranes, which could include the membrane of an organelle or the membrane of the entire cell. You know about the phospholipid bilayer with sterols interspersed to improve its integrity and reduce its permeability. You know about multiple types of proteins and their functions, and you know that these carbohydrate chains are called glycans. To review, you have learned about phospholipids and sterols that together form a bilayer that comprises the main structural component of plasma membranes. Because of their special chemical properties, they arrange themselves in such a way that creates a hydrophobic core, reducing the permeability of plasma membranes, the benefit of which is that an internal space can be maintained that is distinct from the external space, aka maintaining homeostasis. The chemical properties of phospholipids and sterols also mean that the plasma membrane has hydrophilic surfaces that allow it to associate with aqueous solutions both on the inside of the cell or membrane-bound organelle, and it also allows the cell to live within an aqueous solution. You learned about 3 types of plasma membrane proteins: integral, peripheral, and glycoproteins, each with their own important functions. And you learned about carbohydrate glycan signaling flags that are attached to proteins or lipids in the plasma membrane. And you learned about the function of these glycans, which is signaling identity and conveying information.